Hello, everyone, and welcome to today's edition of the 3D Regulatory Genome. It's my great pleasure to welcome Dr. Darío Lupianez today. And I'll give a quick background. Darío has received his master's degree and PhD from the University of Granada under supervision of Professor Jimenez and Burgos, and focused on sex determination and gonadal development in mammals. And that, I think I can say, is still a key interest today for you. Then he joined Professor Stefan Mundloh's lab in Berlin at the Max Planck Institute for Molecular Genetics, where he investigated 3D genome organization and its impact for human implications for human disease. And among other things, there's a very famous paper from 10 years ago now, 2015, that is cited a lot and is one of the seminal contributions to the field still today. And then in 2017, Darío established his own group initially in Berlin at the Institute for Molecular Systems Biology at the Max Delbrück Center for Molecular Medicine, and then relocated to Seville, brought his lab back to Spain in mid-2023. And yeah, and we'll hear about the hang on, continue to share. We'll hear about the evolution of 3D genome organization in Metazoan species today from Darío. Very excited and look forward to your talk.
Okay, so I'm going to share my screen here, there. Okay. Is it working actually?
I don't see it yet.
Okay, so try now.
Yes. Yeah.
Okay. Yes. Now, 1 second. Pointer option. It's a pointer. Good. Yeah. Thank you, Stefan. It's really a pleasure to be here. It's a while since I don't give a virtual talk, so it's let's see how it goes. I hope everything goes fine. So today maybe I'm talking about something a bit different, which is the evolution of 3D chromatin organization, or probably the mechanism that contribute to that. And this is something that we have a great interest on, and that we have been working in the latest years. So I'm very happy to share our recent data on that. So yeah, I think in general, we are really fascinated about this, funky thing, right? Which is, how do you actually fit 2 meters of DNA in the nucleus of a cell, which is, 100 times smaller than a pinhead, right? Of course, you don't need to be very creative to find solutions on how to do that. So you just need to pack everything, right? But what is really fascinating is how can you achieve such a level of compaction and at the same time have a functional genome, right? So this is really what drives our research. And for the latest year, we have been focused on one specific layer of chromatin organization, which are topologically associating domains. And I don't think that I don't need to give that much introduction about it. I mean, we know that TARs are basically, they were detected by using chromosome conformation capture technologies, in particular this genome-wide variant, which is high C. And people started to apply this to a different model organism, and many different labs worldwide, they actually reported the existence of this structure, right? And they basically reflect, large genomic regions, which in vertebrates are around 800 kb average, that actually they seem to be kind of isolated from each other by these regions, which we like to call boundary elements, and they seem to reflect pretty well regulatory landscape, basically where enhancer elements, exert their action over their target genes and give rise to this specific gene expression pattern that we see during development. And Stefan said, we and others, around the mid-decade of the 2000, we actually reported that that's are functionally relevant, and for example, in the group of Stefan Muller, where I did my postdoc, we basically studied this in the context of congenital diseases, but it has been also studied by cancer. In cancer are and now in many different diseases. And there we reported that, basically the disruption of TATs or specifically of their boundaries can actually kind of create novel regulatory interactions, right? And basically fuse, for example, domains, and this domain can basically mix their regulatory content, and this can lead to basically aberrant gene expression, right? And produce different patterns of gene expression. And I think it's at the time we all got very, very excited about this because we thought that we have this is really a fundamental mechanism of gene regulation that it's gonna basically to explain everything, right? And that we can apply pervasively across the genome to really investigate genome function, right? But of course, everything was not so simple because later, many different groups basically they develop strategies to deplete TATs, and they actually saw that basically transcription stayed more or less all the same. So the changes that they reported they were actually minimal. Although it's fair to say that most of these experiments have been done in vitro models, right? But still, it was a surprising result because we expected that we'll remove TATs and everything will go completely crazy, right? And then other groups also reported that insulation is not really absolute. So these are, I mean, these boundary regions seem to constrain interaction across both sides, across TATs, but this doesn't seem to be absolute. So there are basically interaction that can go over that boundary. So this really didn't seem to be a kind of a model to explain everything, right? And I think that nowadays the controversy is still present. So we are still scratching our heads a bit and trying to elucidate what is really the function of that. So I'm not sure I can provide an answer for this in this talk, but maybe I think it's interesting to perhaps reformulate this question, right? And make it a bit different, right? So instead, why don't we ask, which functions might the existence of TATs enable, right? And of course, to address this question, I think it's important that we might approach it through the prism of evolution, right? So basically, some time ago we started exploring this, and we wanted to see if TATs are actually relevant during evolution, and particularly in the emergence of novel traits, right? And for that, we actually choose a pretty extreme phenotype, which are the ovotestis in female mode. So what you can see here, is a picture of a mole. You might be familiar with this animal. It's a cute animal that lives on the ground and makes their nets and he digs and make holes and tunnels, right? But what you probably don't know is that the moles are pretty special because the females of the moles, they are intersexual. So what does this mean? That instead of developing normal ovaries, they actually develop kind of a gonad that it's called ovotestis, and it's a mixture between an ovary and a testicle, right? And what happens is that the ovarian portion, it support sexual reproduction. So the female moles, they actually mate with male moles, and the male moles are, basically very similar to any other mammal. But what happened with this ovotestis is that also has a testicular portion. The testicular portion doesn't produce gametes, but what it actually does is that produce huge amount of testosterone, which is the male hormone. Basically, this is a kind of an exception across mammals. And some time ago, actually, this is a project that was led by Francisca Martinez Real, Fanny, how we know her, and she actually started her group recently here also in the south of Spain. So what she wondered is, how does this happen mechanistically, right? So she studied this from a different perspective, like studying the transcription, regulatory elements. She could not find anything. And then, we had the idea that maybe, there could be a chance that there are something involving that, right? But before we go to that, so we have to say that the life of a mole it's really tough, right? I mean, imagine that you need to be your constant life, like digging tunnels in the soil. I mean, this is physically very demanding, but also you need to be fighting against all the different small animals that are underground and actually competing for very scarce resources. right? So actually, if you live in this environment, there is something that could be very beneficial, which is actually to have more androgens or the male hormones. So why is this? Because if you have more androgens, generally you will develop more muscle, so you will be stronger, and this will give you a selective advantage to dig tunnels. And also because an increased amount of androgen has been reported in many mammalian species that it's linked to aggressiveness, right? So if you have that and you are become more aggressive, then you can fight more efficiently for resources. So what I did not told you about the moles is that they are super solitary animals, right? So actually they have a breeding season where the male mate with the female, and then after this the male goes away, and the female carries the burden of raising the pups, right? So if you think about this, because they are very solitary animals, of course, there is a clear disadvantage, because the males have the hormones, so they become stronger and more aggressive, and then it could be that the female have a bit of disadvantage. So what evolution did is that it actually equalized the strength among the sexes in the moles, and this, of course, is linked with the ovotestis, because the ovotestis is the organ that is actually producing this huge amount of hormones that I told you before. So how did we study this? So actually inspired on our previous research, so we developed a comparative 3D genomic strategy that tries to identify genomic rearrangements that basically disrupt that, right? And basically that create new regulatory configurations, right? So the idea is that, we will do sequence comparison at the level of synteny between species that have the trait, the mole, and other species that do not have it. And then we will try to identify synteny breaks, which actually reflect regions of the genome that have been, broken or that have a different configuration. And then on top of that, we will analyze Hi-C data to see if these synteny breaks have basically rearranged a TAD in a specific species, in this case, the mole, and this might have put some regulatory elements in contact with new genes, and this might give rise to, a specific pattern of expression. So, cutting the story short, because this is published a while ago. So what we found is that at the FGF9 locus, and this is a very interesting locus because, it's a 2 megabase TAD, where FGF9 is the only protein coding gene, and this is actually a protesticular factor. When you have expression of this, you basically promote testicular development. So what we found is that basically there is a synteny break at the end of the FGF9 TAD, and this is the product of a 26 megabase inversion that is specific of moles. So it's not in other mammals, right? So basically what happened in the mole is that this part of the TAD, it goes away, and something completely new comes here, right? And what we saw, I can tell you this basically, so that this region is actually enriched in regulatory elements that are active specifically in the gonad of the female mole. So you can already imagine what might happen, right? And what you can see here, it's basically the expression of FGF9 in the mouse, right, that doesn't have this rearrangement and do not develop ovotestis, and you can see that the expression pattern, it's clearly dimorphic. So you have more expression in the male compared with the female. So in the mole, basically the level, they stay the same irrespectively of the sex, right? So in a way, this genomic rearrangement is actually associated with an increased expression of FGF9. So we actually wanted to prove this functionally, and what we did is that we basically took a mini gene for FGF9, and we embedded this in a bag that contains the regulatory regions of WT1. So WT1 is actually a gene that it's normally expressed during ovarian development in the mouse. So we are kind of recreating the pattern of expression of FGF9 in the mouse as we have seen in the mole. So with piggyback transgenesis, we inserted this construct into the mouse genome, and then we analyzed female transgenic mouse. And what we saw, it was actually pretty striking, because what you can see here, it's a wild type ovary, where you can see the ovarian follicles here distributed, and when we overexpress this gene, similar to what we have observed in the mole, we see the partial transformation of this gonad into something that actually resembles an ovotestis, with the testicular tissue here completely devoid of germ cells, as we see in the mole, right? So this is interesting because it's I mean, as a small summary of this initial part, we actually found that the stat rearrangements might lead to the emergence of novel traits, and basically very similar to the mechanism that I showed you before for human diseases, right? So the mouse is not the only species that we studied. We also have a study in 2023 where we actually studied another pretty striking trait, which is the enlargements of the fins of skates, which they actually employ them, to swim and to hide for camouflage, from predators. And there we actually found something very similar. It's a big rearrangement that creates a new regulatory unit by rearranging the TATs, and in this case was for prickle-1, which is the main effector of the PCP pathway, which is involved in tissue growth and polarity, right? Okay, so that was interesting. But then, we actually wanted to, get a bit more insights on TATs, on what is probably the evolutionary relevance of them, right? And we make a very simple question, are actually TAT structure? I mean, can you observe across Metazoan species, across animal species, right? And actually, so Rafa, who was a former postdoc in the lab, now also establishing his own group. So we basically, at that time, we took the whole phylogeny, we tried to evaluate every high C experiment that has been done, and we tried to put a bit of sense in the Metazoan phylogeny, right? And so basically, here on the top, you can see mammals, and then you go down, you can see vertebrates, your vertebrates, chordate, the pterostome, here, invertebrates, and even plants, right? And I think, I don't really need to explain this. Everything looks very visual, right? So in gray, you will see the ones for, at that time, we did not have evidence of TATs having, and basically what you can see is that TATs are everywhere, right? So they are really pervasive, and even, this type of structure have been actually reported in plants, right? So they seem to be basically everywhere. So, but what do we know about TATs? So how they are actually formed? So I think in the field, it's pretty well accepted that, at least functionally, that seem to be formed by a process of loop extrusion, right? So basically, you will have the chromatin fiber, and on top of that, you will have the cohesin complex, which have a ring shape, and then the cohesin complex will actually go on top of the chromatin, and it will start basically extruding and creating those loops, right? And the process of loop extrusion seems to be stabilized by an insulator factor, which is called CTCF, right? And then it will kind of stabilize these loops, and this will basically give rise to the very specific shapes that we see in Heisey maps, right? So, basically we will have a TAT here, and the TAT normally have a focal point of interaction at the apex, which kind of depicts kind of a stabilized interaction between the boundaries of a TAT that normally are enriched in CTCF. So, okay, so we know about this. So perhaps, the question that we were doing before is not the correct one, but it's more, it's not that if TATs are pervasive or conserved across animal species, more if the mechanisms that actually form TATs are the same, right? And here is where things become a little bit more interesting, right? So what we know from the mouse is that basically the patterns that emerge from a Heisey map are the interplay between two different mechanisms, right? So one of them is loop extrusion, which is basically forming the dots, and you can see them here, like for example here with the focal point of interaction, right? And then there is a second mechanism, which is called compartmentalization, which is a bit kind of hidden behind the dots, right? So the dots seem to be a kind of a bit more prominent in that sense. And compartmentalization is basically, the interaction with regions that are basically homotypic, like, for example, regions which have an active epigenetic status, they like to be with each other more frequently, and the same can be said for regions that are more inactive, right? So what happened, for example, when we go to another animal model, when people started to do high-C on Drosophila, they also report the existence of TATs, but when the resolution of this high-C experiment kind of increased and increased, they actually realized that the signal was not really the same. And what you can see in Drosophila, or the pattern that you can see, they actually correspond more with compartmentalization, this type of homotypic interaction. And this is because this type of signal, um, in contrast to the signal that we see that it's kind of associated with the TAT, they seem to give a more like a plate pattern, like a bit more like a chessboard checker pattern, where you will have, for example, this domain here that will interact with this large domain here, and this is the square of interaction, and in between you will have these kind of small domains, right? So the question now is that I told you that, for example, CTCF, it's kind of very important to create this pattern because it can really alter the loop extrusion process, right? And the interesting thing about this is that when you knock out CTCF, you basically get completely devoid of TATs, and then this compartmentalization signal emerge more. And the interesting thing about this is that when you knock out CTCF, so this is little developmental. I mean, not only developmentally, you cannot even culture cells if you don't have CTCF, because the cells will eventually die, right? But this really contrasts with the effects of knocking out CTCF in Drosophila, because when you knock it out, the effects on 3D chromatin organization are, I would say, more moderate. Most of these signal is retained. I mean, basically because this is more associated with compartmentalization, and only at very selected loci you can see that there are prominence effects associated to the loss of CTCF. But I think that the most important thing about this is that so CTCF, it's basically dispensable for early development. I mean, you can remove it and then you will form an embryo. I mean, you will see some developmental defects in the adult, but you can progress through development without CTCF, right? So if we go back a bit, I mean, this actually creates a very interesting question, because what it's really a TAT, right? And for this, I always like to cite, I would say, a legendary tweet from Victor Corses that says that everything will make more sense if people will acknowledge that triangles at diagonal touch is a visual and computational concept that it's devoid of biological significance. I think that he really hit the point here, because it could be that you see a signal that looks like a TAT and you analyze it, but the problem here is that the TATs were discovered based on a computational concept. So they were discovered before we discovered the mechanisms, and we tend to put everything under the same definition. But if you really don't understand the mechanism by which these structures are formed, you really don't understand the biology of this. So now if we actually come back to our tree, what we know is that there are a class of TATs, which we can call loop extrusion TATs, that seem to be dependent of CTCF, and then there are other TATs that are independent of CTCF, right? And this is pretty interesting. So for the rest of the talk, please keep this in mind. When I talk about TATs, I will be referring only to these TATs, so the ones to be formed by loop extrusion. But this is really interesting because, CTCF, it's basically a protein that emerged in bilateria. So in all these different species, even in the ones that form TATs independently of CTCF, so CTCF is mostly present, right? So that actually points to the fact that CTCF protein might have evolved different functions, right? So this is actually what we became really interested in, and something that I studied in collaboration with Robert Simpson in a project that was led by Liene, and also with the help of Rafa. So basically what we wanted to do is to see if the ability of CTCF to contribute to the formation of the TATs by loop extrusion is intrinsically encoded in the protein, right? And for that, we basically had in mind a very, I would say, crazy experiment, right? So what will happen if we will have a background where loop extrusion can happen? So basically, you have all the ingredients, like for example, a mouse embryonic stem cell, and then you put the different versions of CTCF of different species where we do not have evidence that TATs are formed by loop extrusion, or that CTCF has any function on that, right? So basically the idea was, to take mouse embryonic stem cell, get rid of CTCF, and then put the CTCF from a completely different species. But I told you before that the knockout of CTCF, it's lethal. So you can basically, you cannot culture the ESS, right? So what we did for that was basically a system that it's basically, that it's based on the one that Fresh Nora employed to investigate the function of CTCF. So it's this classical auxin diagram. So what we did is that, so we basically modify the CTCF locus, and we put, the diagram tag here with a GFP in the two alleles. Everything goes in the Tigre locus. And then one of these copies, we actually substituted by a triple flag orthologous from a different species. So, in one of the alleles, you will have the endogenous contribution, okay, that will keep the cell alive. In the other, you will have constant firing of the orthologous from a different species, right? And at some point, we will add auxin, so we will get rid of this, the endogenous one, and only the one that is specific of a species will remain expressed. So you can see here a Western blot where you can basically see that we cannot detect protein of CTCF after we add the auxin. And something that it's really important is that in this CTCF, we only put the coding sequence from a different species. So we are basically preserving the endogenous promoter from the mouse, the 5' UTR and the 3' UTR, so that we can be sure that the effects that we see can be attributed to the sequence of the protein. So we basically did this with a bunch of species, right? So the first one that we did was kind of the mouse in mouse, so this is our control. We also did it like putting the protein of zebrafish, or putting the gene of zebrafish, which is a species, it's a kind of a distal vertebrate to the mouse, and there are evidence that, compartmentalization and loop extrusion coexist in this species. And then we put a bunch of invertebrates. So a distal chordate, which is Siona, the sea squirt, then a distal deuterostome to the mouse, which is the sea urchin, and then basically the fly, as the more distant species. Here you can see that, our triple flag from the different species is basically, generating a protein that can actually be detected with the same antibody, which is a flag antibody. And you can see here different sizes because, the protein has different sizes in the different species. I want to draw your attention to, because, we have seen that we obtain a lower level of protein for the more distant species. We know that this has something to do with codon optimization, but we were not that much worried about this because, it has been shown that even CTCF levels that are almost undetectable in a Western blot can rescue 3D chromatin organization. So we reason that, if we have the protein expressed at this level, I mean, if there is some effect to rescue, it should be rescued already. Okay, so what did we do next? So we put the auxin, we did Hi-C, and we try to see what happens. So here you can see the Hi-C map for the SOX9 locus in the mouse. So this is our positive control, mouse on mouse. And you can see here our nice stats, okay? What happened when we put, or when we start to express only the protein for Siona, sea urchin, or the flies? The tats are completely gone. Okay, I can tell you that there is compartmentalization, but you cannot see it at this resolution. So there is a bit of residual structure, but the tats are completely gone. And this is another way of visualizing this, because this is the insulation score. And every time that you see a minima, it will actually correspond to the position of a boundary in our control, which is in gray here. And you can say that we almost get a kind of a flat line without not signal. So the insulation, it's almost gone. And you can also see this here with the boundary score distribution. You can see that in the mouse, we have basically this range of boundary scores, and everything is basically shifted to a lower boundary score. And this is actually, kind of overlapping with the profile that you will have if you will remove CTCF completely. So the tats are gone. Okay. What happened when we put the zebrafish? I mean, they actually look like, mirror replicates. So it's pretty astonishing. I mean, you cannot even differentiate them from, the two biological replicates of the mouse. So, the tats are basically in the same position, and the boundary score that we calculated, they are basically the same. So despite more than 400 million years of evolution, the function of CTCF seems to be pretty well conserved across vertebrates. Okay. I would say that this might be a result that one could expect. But here is where things get a bit more interesting. So I told you before that so we have a flag tag, right? And that we could actually detect the protein with the same antibody. And I think that this is really powerful because then we can do chip-seq experiments in all the different mouse CSS that we have done with the different replacements and obtain a signal that it's really comparable because it's basically all done with the same antibody. So when we did these experiments for mouse and for zebrafish, you can see that again, the profiles of binding for CTCF, they are again, they seem to be kind of mirror images. What happened when try to detect from invertebrates, I mean, we see some signal here and there, and which, it might look like residual at first sight. But we were, very puzzled when we actually saw that in Siona, this basically in Siona robusta, we see that there is kind of pervasive binding of CTCF, right? And we were intrigued by that. So what you can see here, it's basically so we make a peak calling with all the different 5 samples, and we basically plot here the chip-seq intensity signal in every one of these regions that we have basically detected across all 5 species, and we basically make a clustering, right? So what you can see is that we have a kind of a first cluster, right, which is basically only specific of vertebrates. Okay. Then we have a third cluster, it's basically including vertebrates plus Siona. Then we have a third cluster, which is basically that Siona binds more strongly, but also you can see binding of vertebrates. And then we have a fourth cluster where all of the proteins from the different species seem to bind, right? And here what you can see, it's basically the insulation score associated to each one of these regions. So you can see a very clear pattern. So here are the 2 species that basically retain the tats. And you can see that, they have, so the blue signal is way more prominent compared to the ones that do not retain the insulation, which actually these 3 correspond to the invertebrates. And you can see that the signal is almost identical to the one where you do not have CTCF. And this is pretty interesting because, if I draw your attention here, which is, those events where Siona seem to be also binding, the binding of Siona is not able to induce insulation, right? So it seems that, I mean, you need to compare this one with this one. So it seems that, you can bind the protein here and there, but, the rescue effect that we're looking for and the formation of stats, it's not really there. Okay, so what we can do with this is that, we can take each cluster and we can try to perform a motif discovery with the peaks, right? And I thought that, here we can see that some patterns start to emerge. Like, for example, when we do this with cluster 1, which is the one that is specific of vertebrates, we actually retrieve the CTCF motif that has been previously described for vertebrates. This is kind of this very long motif of around 29 different nucleotides, right? Then when we actually move to cluster 2, we see that the motif it's pretty identical. It's actually almost the same for cluster 3, right? The model looks pretty identical, but there is a residual C here, which is not present, when the vertebrates are actually binding. And then with cluster 4, we actually retrieve this truncated motif that has been described for Drosophila before. And I think it's pretty interesting because what we see is that, there is this terminal C here, which seems to be specific and essential for the invertebrate protein to bind. And I think that this is pretty interesting because if you have an additional C, it looks like you are creating additional chances for creating specific methylation of cytosines, right? And we know that this is important because CTCF binding it's pretty sensitive to methylation, right? So in a way, it looks like there might be this kind of evolutionary conflict, right? On one hand, you have this C that seems to be essential for the invertebrate protein to bind, which is not really present in invertebrates, right? And this is interesting because invertebrate this type of methylation it's a really prominent mechanism which is actually not happening that much in invertebrates. If I just move to this slide, we actually confirm that this additional C is susceptible to undergo methylation. Because what you can see here is basically, we look in the genome for all instances of the invertebrate specific motif, and then we plotted on top the signal of ChIP-seq. So you can see that we basically have two very big clusters. So the ones that are basically binding CTCF and the ones that are not binding CTCF. And here we actually went to dig and to get methylation data from mouse embryonic stem cells. And you basically see this line here that actually depicts that most of these sites are actually methylated. And if we actually zoom in, many of them, I mean, you can see that you have signal here, but basically the signal is spread all over this part. It's specifically methylated at this terminal C that it's specific of invertebrates. So there is really a clear correlation between the fact that this cytosine is methylated and the absence of CTCF binding. And if we come back here, so what I wanted to say is that it's not only that we found like different motifs of binding. Things also become very interesting when we actually look at the genomic distribution of these binding events. Because what we see is that for cluster 1, most of these bindings, they actually tend to be associated to regions that are basically distal to the promoter. But so approximately in cluster 1, we see that 25% of the events are very close to the promoter. But this tendency is completely shifted, when we go to the clusters that involve invertebrates, and particularly for cluster 3 or cluster 4. So here, most of the bindings seem to be near the promoter and not so distal, right? And I think that it's also very interesting to think of this in terms of evolution, because what we can say is that vertebrates, they tend to have larger genomes compared to invertebrates, right? So, one could be a bit speculative here and try to think that maybe CTCF could be actually involved in the regulatory expansion of the genome that is very characteristic of vertebrates, right? And that might be actually supported by this shift of binding towards more distal regions. Okay, so if we actually come back to this binding profile, there is something that got our heads really scratching, and was the fact that the Sionna protein seemed to be binding, in almost half of the sites where the vertebrate protein was actually binding. So we thought, it's really surprising that we do not see any major effect on 3D chromatin organization, right? So, but we wanted to investigate if this could mean something. And what we did before, so I told you before that the experiment that we did was with this oxyintegron, and there was a reason for this. So we reasoned that if we put a protein there that cannot actually rescue the function of CTCF, the cells will basically die, right? I don't know. We felt a little bit brave about these results, and we say, okay, what will happen if we will do a homozygous substitution? So we will remove completely the protein of mouse, and we will just put 2 copies of the protein of C squared and just leave the cells there to see what happens, right? So this is what we did. So we did a homozygous direct replacement, and we did it for 2 of the orthologous. So one of them is the one from the sea urchin and from the C squared from Sionna, and the other from the fly, right? And we were very I don't know, surprised to see that for the fly experiment, we could not retrieve any clone that has the integration on homozygosity. All the ones that we could retrieve were either wild type, so they did not have any integration, or they have the integration in heterozygosity. But for Siona, we got up to 40% of the clones that we retrieve with homozygous integration, right? And you can basically see it here. So this is basically a Western blot that it's recognizing the CTCF protein in the mouse control and in our homozygous line for the Cs for Siona, right? And you see that they basically have no expression of the endogenous CTCF protein of the mouse. They only have the expression of the one from Siona. And what we saw is that, these cells, they actually live. I mean, they are fine. I mean, they are not super happy. So you can see that the proliferation goes a bit down, but you can passage them and they will not die, right? And I think that this is really cool because, it actually shows that CTCF has really two different functions, So one of them, I'm talking about the vertebrate CTCF protein, right? So one of them is probably to mediate 3D chromatin organization, and another one that seemed to be also shared, at least with Siona, it's a function that we think it's probably more like a transcriptional regulation, which is the one that it's basically rescuing the viability of mouse embryonic stem cells. And I have to say that, we are not the ones to really propose that CTCF have more functions beyond 3D chromatin organization, but we are excited because, this is the one proof that you can actually uncouple these two different functions in experiments, and also that these two functions have been also uncoupled, in the context of evolution, right? Which I think is pretty exciting. Okay, so I basically show you that the CTCF version of vertebrate, it's actually necessary to form the TATs, right? But is this vertebrate CTCF sufficient to form TATs? So to address this question, we did kind of the opposite experiment. So we took the CTCF from a vertebrate, so the sequence of the mouse, and then we put it in a background where we do not have evidence that TATs are formed by loop extrusion. So that's the fly, okay? So this is basically the experiment that we did. This is a bit more complex than the previous one. So we basically modified the CTCF locus of the fly to put a triple flag and a recombination site, an FRT site here. And then we modified it again to basically put another construct here downstream. So this construct has the mouse CTCF flanked by an FRT. And here we have a GFP that it's basically firing the GFP normally. So when you cross the flag line with an FLP line, what you get rid is basically of the CTCF from Drosophila and also of the green fluorescent marker, right? And only the mouse CTCF will be expressed together with the flag tag, again, to do chipsec with the same conditions. Here are just some controls. So, before crossing with the FLP, you can see the larvae, which are basically expressing the GFP, and then after the FLP, you basically get rid of the GFP and also of the endogenous protein. And here you can see a chipset with the flag, where you can see, basically that we are expressing the mouse CTCF, and also this is another line when we did the fly-on-fly experiment as a control. So you can see that you are only expressing the CTCF version that has the flag on that. Okay, so we did Hi-C, of course. What is the result? So that the presence of the mouse CTCF is not able to induce the formation of TATs on a fly background. So it seems that you might need to have something else to form this structure, right? So what you can see here, it's basically the insulation score. You can see that for most of the boundaries, the insulation score remains the same. We only see, kind of minor changes in a subset, and these are basically the same loci that have been associated with disruptions of structure in in a previous publication by the group of Maria Gambetta, where they get rid of CTCF. So you can basically see, you have this insulation here at this boundary. We put the mouse protein here. This insulation is gone, and the same happens when you don't have CTCF, right? So it's not only that CTCF, the mouse CTCF cannot form TATs by loop extrusion, at least, or equivalent to the one that we see in vertebrates when you put it in the fly background, is that it's not even able to perform the function that the fly CTCF is performing in the flies. And we see this because the knockout of CTCF has a very characteristic phenotype, which are some abdominal defects and also on the genital region, which are also present when we put the CTCF protein from the mouth there. Anyhow, the surprising thing again was that the CTCF protein of the mouse is binding, quite prominently in the fly genome. So you can see that there are many peaks here and there, right? And here it's basically I'm showing you we are following the same strategy as before. We have the two different sets of chip-seq experiment. We make peak calling, and then we basically plot the signal and cluster according to the signal. So we have now 3 very prominent clusters. So one of them is only specific for the fly, another which seem to be kind of shared between both orthologs, and another that seem to be kind of almost exclusive of the mouse protein, right? And then, when we perform the motif discovery, we're very glad to see kind of the equivalent picture to our previous experiments, right? Because we see that this C that seems to be particular only of the binding of the invertebrate protein it's also still there, and it's completely absent of the cluster 3 of CTCF. Moreover, we also see, the shift in the ratio between distal and proximal to the promoter, right? So in the cluster 1, where and that is specific of the fly, most of the binding is near the promoter, and this is completely shifted, when you put the mouse protein, where most of the binding is actually at distal regions. So with that, we are reaching an end. As a summary, I show you that the vertebrate CTCF are functionally replaceable despite vast evolutionary distances. That CTCF function, they can be uncoupled. So one of them as an architectural protein and another as a transcriptional modulator. That the version of CTCF of vertebrates is actually required but not sufficient to form TATs, and that CTCF has shifted its binding distribution and motif preference during evolution. And really to close, I would like to come back to my previous question. Which functions might the existence of TATs enable? So in my opinion, I think that one of them it's kind of modular evolution. You will have these domains and this kind of regulatory domain that will be relatively isolated, from the influence of other regulatory activities, and they might be shuffled around so that the regulatory content can be mixed up and create new pattern of expression. So I think it's interesting to think about that also as evolutionary units, and really the work by José Luis Gómez Escarmeta and Nacho Maeso and Rafa Zemer was really pioneering, because they already put this concept there in 2017, and in a very beautiful piece of opinion that they wrote. And I think of a second function that is probably related to this promoter and distal shift in binding. It might relate to regulatory expansion, right? And what I had commented before, that in vertebrates, the genomes tend to be larger, so you can accommodate more regulatory elements, and of course, kind of a mechanism that allows you to perform loop extrusion, right? It allows you to basically to reach for regions that are more distal, so you can potentially interact with more regulatory elements. And I think it's the level of compartmentalization that's also provide, although it's not absolute in all the cases, they also help to facilitate this pleiotropy, because if you increase pleiotropy, you also need a mechanism that puts a bit of control that everything doesn't go completely crazy. And I think that this really align well with recent experiments or paper from the lab of Luca Giorgetti, Wouter de la, Wendy Bigmore, many others that have actually shown that loop extrusion, it seems to be particularly important to support distal regulation and also at developmental loci like sonic hedgehog. So with that, I would like, of course, to thank all the people of my group. As Stefan mentioned, we came back recently to Spain, and we are happy, to be back home and to have such an exciting group. And also, of course, the collaborators, funding agencies, you for your attention. And of course, I'm happy to take questions if you have. Thank you.